Next one is Human Genome Project. Human Genome Project is nothing but human total number of genes identify பண்ற ஒரு project. What is meant by genome? Genome is nothing but a complete set of genes present in a cell. ஒரு single cells approximately 20 to 25,000 genes are present, and 25,000 genes, how the genes are arranged in a cell, in a DNA, how the base pairs, for example adenine thymine cytosine guanine, adenine thymine cytosine guanine, adenine thymine cytosine guanine என்னோட செல்ஸ்ல இருக்கு. But in your cells adenine thymine, adenine thymine, adenine thymine. In some other person's cell cytosine guanine, cytosine guanine, cytosine guanine. In some other person's cell guanine cytosine, guanine cytosine, guanine cytosine. So the various person cells which contain different arrangement of base pairs. In this arrangement 99.9% of DNA arranged sequentially same in all the species. All the species means all the person in a species. For example Homo sapiens அப்படிங்கிறது ஒரு species. Homo sapiens எல்லா human being லையும் 99.9% of base pairs are arranged same arrange ஆயிருக்கும். But 0.1% மட்டும் difference தான். So அந்த 0.1% difference only make the human DNA in this distinct features. ஒரு பர்சனுக்கு இன்னொரு பர்சனுக்கு நிறைய டிஃபரன்சஸ் இருக்கும். இந்த டிஃபரன்சஸ் ஆர் டியூ டு அந்த 0.1% DNA base pairs. ஸோ எப்படி இந்த 99.9% of base pairs are similar in a DNA, in a structure. ஸோ அப்படின்னு சொன்னோம்னா the particular தான் Human Genome Project. It is actually a mega project. It started in 1990 ஸ்டார்ட் பண்ணாங்க. US and Russia and various countries contribution 1990 Human Genome Project started. But normally we know that is aims of Human Genome Project is actually human haploid content 3.3 into 10 to the power of 9 base pairs are present, and 3.3 into 10 to the power of 9 base pairs என்ன ஆர்டர்ல அரேஞ்ச் ஆயிருக்கு. இப்போ என்ன ஆர்டர்? adenine thymine, adenine thymine, adenine thymine, adenine thymine, adenine thymine, or cytosine guanine, cytosine guanine, cytosine guanine order, or maybe guanine cytosine, cytosine guanine, guanine cytosine, adenine thymine, thymine adenine, adenine thymine. So how the DNA base pairs are arranged in a particular gene, or how many genes are present in a human cell, ஒவ்வொரு ஜீன்ஸும் என்ன ஆர்டர்ல அரேஞ்ச் ஆயிருக்கு in the base pairs. So அந்த total information gather பண்ற ஒரு ப்ராஜெக்ட் தான் Human Genome Project. It's already over. That is a 13-year mega project. It is in the first day, the first 1990 இந்த ப்ராஜெக்ட் ஸ்டார்ட் பண்ணும்போது the project cost US டாலர்ல $3 per base pair. So ஒரு base pair ஐடென்டிஃபை பண்றதுக்கு in olden days in 1990 $3 approximate செலவாகும் அப்படின்னு கால்குலேட் பண்ணிருந்தோம். Then approximately in the 3.3 into 10 to the power of 9 base pairs calculate பண்ணோம்னா $9 billion வந்து is செலவாகும் அப்படின்னு ஒரு calculation செலவாகும். Then suppose the DNA base pairs print பண்றோம் inside the book அப்படின்னு. Inside the book means the book contain, the page contain 1000 letters, 1000 pages இருக்கிற ஒரு bookல இந்த information like adenine thymine cytosine guanine எல்லாம் print பண்ணினா you need 3300 books for the storing information a particular cell. ஒரு particular human cells இருக்கிற base pairs நீ print பண்ணி வச்சோமா, so இவ்வளோ பெரிய books தேவை. 3300 books are needed for storing this information. Okay, so it is a multi-crore project. This is next in the particular Human Genome Project the new evolution, new discipline of biology, that is a new branch in biology, that is bioinformatics is developed. Bioinformatics means biology plus information science, because without these books, without this any system or language or code words, we can't easily check the particular genome, particular genes base pairs. with the use of software only, with the use of language only, with the use of system knowledge only, the computer, modern computers only, we identify, redrive and store and redrive the information from the system. It is a easy work. Otherwise, books it is so tough to calculate or to analyze the human genome. Okay? Yes. Next one, goals of the Human Genome Project. The use approximately human cell contain 20 to 25,000 of genes. For identifying 20 to 25,000 of genes, the Human Genome Project is important. Suppose we identify the genes means, suppose if any modification in genes, genetic disorders like sickle cell anemia, hemophilia means, we cut the defective gene in embryo stage and correct the particular gene means the genetic disorder is cured. So, for this information, for this disease management, so Human Genome Project is important. First we identify the genes, then only we correct the defective gene. So, which gene is correct, exactly correct, and which gene is defected. First we analyze the data of gene only, we conclude this which gene is affected, this is genetic disordered affected gene, this is sickle cell anemia affected genes. Then only identify and correct it with the use of gene therapy. For this gene therapy, suppose the Human Genome Project is important for identifying all the genes in a cell. And the 3 billion base pairs are present. We know that the haploid content of human genome, that is 3.3 into 10 to the power of 9 base pairs, approximately 3 million base pairs are present in a human cell, that identifying the particular. Then store this information in a database and tools for data analysis, because this is actually a mega project. Suppose we are stored in printed in books means, we analyzing the particular gene, it takes long time because 3,300 books is needed. So, how we analyze the 3,300 books, refer the 3,300 books, in each book 1,000 pages also, how, and we take a particular information about the particular gene with the use of system knowledge only, system language only, modern system only, easily we transfer the information to the other sectors, or maybe analyzing the tools and analyzing the DNA, and with the use of gene analysis the information is transferred to the another sectors. Another sectors means like industries, agriculture, other sectors also idu nam analyze panna mudiyum. So, otherwise this is other sector. For example, how genes is defected, then how we correct the particular gene with the use of biotechnology, the use of microbiology, totally we modify the gene. So, other sectors the information carry over. Then for analyzing the human genome, we receive various complaints from various sources like ethical, legal and social issues also there, because enoda genes ni express pannina enoda characters ella illa therinjirum. Suppose social issues, ethical issues, legal issues are also awaken, so it arises. So, on the ethical, legal, social issues, that is LC, ethical, legal, social issues, other analyze pandradhukku some committees are present. And it is actually a 13-year project, not a 1 or 2 years project. It is a 13-year project. After 13 years only, various discipline scientists, various branch scientists like physicist, chemist, biochemist, biologist, microbiologist, biotechnologist, computer software engineers, in the field of various scientists, join panni the Human Genome Project complete pannirukanga. So, it is a mega project for analyzing a single cell genes, base pairs, how the base pairs are arranged, in what way, in what order they are arranged. That is Human Genome Project. Okay, it is useful in DNA fingerprinting, in agriculture, medicines, therapeutic product, gene therapeutic product, various branches. It is useful. Okay. In Human Genome Project, various organisms other than the human, non-human organisms also identified with the use of genome project. So human genome is, we know, it is a 3.3 into 10 to the power of 9 base pairs in the haploid content. But other organisms, if any other organisms DNA is similar to the human, so with the use of research only we identified some organisms genes are some relation with the human genes. So for genetic modification, for the biotechnology, for the gene therapy, we use such organism for transferring the genes from other organisms to the human source. So the particular research also done with the use of genome project, like a bacterium genomes is identified, and yeast total genomes, all the base pairs in the yeast chromosome and all the base pairs in the Cyanohabitates elegans is actually a nematode, a pathogenic, non-pathogenic nematode. And Rhizobium melanogaster, we know that the fruit fly, and rice plant, the genome, and Arabidopsis organisms genome. So various organisms genome base pairs are identified with the use of genome project. Suppose these organisms genes are similar to human genes, means we use such genes for the genetic research and the gene modification and the gene therapy. So other organisms genomes also identified with the use of genome project. And the methodologies how the genomes are identified. So the methodologies is two. First, all genes in the DNA. Normally human DNA contain many genes. Some genes are expressed in the form of protein, some gene is not expressed in the form of protein. Then in first approach in human genome project, all the genes in the DNA we identified. All the genes in DNA we identified and make the gene which is useful for the research. All the genes. Suppose some genes are not expressing the character, some genes only expressing the characters, expressing the protein and producing the protein. All the genes is which are not expressed or may be expressed. All the genes present in the DNA are identified and sequenced with the use of first step. In methodology, two methodology. One method we identify all the genes. In another method, only some genes only expressing the character. So on the genes mattum podula edukku ella genes identify pannum. It is a waste. This is a cost wise time wasting process. நம்ம particular gene எந்தெந்த genesலாம் எந்தெந்த character express பண்ணுதோ அந்த particular genes மட்டும் identify பண்ணா it is useful for our research. So in the second approach என்ன பண்றோம் genetic scientist only expressed sequence tags, the gene which expresses the character, in expressing the polypeptide chain, which expressing the protein. நம்ம படிச்சோம்ல the structural gene which is important for the mRNA and mRNA important for the protein. So அந்த எந்த particular structural gene is expressing the particular character or particular protein, அந்த particular gene மட்டும் identify பண்ணி அந்த genesல என்னென்ன base pairs இருக்கு, order, how the base pairs is ordered, அதை மட்டும் identify பண்ணா that is expressed sequence tag, that is ESTs. In our board exam, in one board exam கேட்டாங்க EST means what? EST is nothing but expressed sequence tags, that is the gene present in the DNA. It is important for the mRNA and the protein production. Some genes only expressing the characters, expressing the mRNA, expressing the protein. And the particular genes are expressed sequence tag. And the particular gene மட்டும் identify பண்ணலாம். So human genome project la ரெண்டு methods. One method is completely all the genes, we identifying all the genes in the chromosomes or in the DNA. In another approach only the genes are expressing the protein, only the genes are controlling the character, only the genes are expressing the characters, and the particular gene மட்டும் identify பண்ணலாம். So on the ரெண்டு approach human genome project is moving. And other than the human also, the bacterial artificial chromosome, yeast artificial chromosome, இப்போ இந்த particular genes எடுத்து human, for example, how we identify the gene is working or functioning or producing the particular protein நம்மளுக்கு தெரியாது. அப்போ particular gene கட் பண்ணி எடுத்து bacterium cell இருக்கிற ஜீனில் வச்சிட்டோம்னா the bacterium cell is expressing the which amino acid which protein means அப்போ அந்த ஜீன் அந்த ப்ரோட்டீனை எக்ஸ்பிரஸ் பண்ணுது அப்படின்னு ஐடென்டிஃபை பண்ணிடலாம். So for this we identify the bacterial artificial chromosome and the yeast artificial chromosome for this human research human genome project. ஏன்னா human genes எப்படி எக்ஸ்பிரஸ் பண்ணுறதுன்னு எப்படி நம்ம கண்டுபிடிக்க முடியும்? With the use of other organism, with the use of bacterium, with the use of yeast only we conclude இந்த ஜீன் இந்த கேரக்டரை எக்ஸ்பிரஸ் பண்ணுது, இந்த ஜீன் இந்த கேரக்டரை எக்ஸ்பிரஸ் பண்ணுது, இந்த ஜீன் இந்த பர்டிகுலர் ப்ரோட்டீனாக ப்ரொடியூஸ் பண்ணுது, இந்த ஜீன் வித் பர்டிகுலர் கேரக்டர்ஸாக பர்டிகுலர் பிக்மெண்ட்டை ப்ரொடியூஸ் பண்ணுது. So that the identification of a particular gene function we need some other organisms like bacterium and yeast. So அப்போ the bacterial artificial chromosome, yeast artificial chromosome இருந்தால் அந்த human genome project is completed. So அதுக்கும் அதை யூஸ் பண்ணுறாங்க, அதையும் கண்டுபிடிச்சிருக்காங்க. Then it's a humanல நம்ம பாசிபிள் கிடையாது for analyzing the adenine thymine. எப்படி கண்டிப்பை பண்ணுறது 3.3 into 10 to the power of 9 base without any errors adenine thymine cytosine guanine. This identification is so tough, is humanly is not possible. So அப்போ some machines, some automatic DNA sequencers, அந்த automatic DNA sequencesனா நீங்கள் DNA உள்ளே போட்டிங்கன்னா அதுவே sequence அனலைஸ் பண்ணி so how the sequence are present, how the sequence are ordered, how base pairs are ordered adenine thymine in orderல இருக்குங்கிறது ஆல்ரெடி அந்த மிஷினே சொல்லிடும். அந்த மிஷின் is known as automated DNA sequencer. The process is automated DNA sequence. With the use of sequencer machine we identify the particular gene setup, particular gene arrangement, particular base pairs arrangement, and the particular base pairs arrangement is identified with the use of Frederick Sanger. Frederick Sanger abdin scientist identifying the amino acid sequence in a protein. So normally we know the protein consists of various amino acids. In order amino acids irukum, we know that the protein, the first amino acid is methionine, that is AUG codon, and how the methionine amino acid combine to other amino acids, how the protein is formed. So one protein, what are all the amino acids are present, அதை எப்படி அனலைஸ் பண்ணுறதுன்னு ஒரு டெக்னிக் கண்டுபிடிச்சது தான் யார்? ஃப்ரெடரிக் சாங்கர். தட் இஸ் ஹவு த அமினோ ஆசிட்ஸ் ஆர் அரேஞ்ச்ட், வாட் ஆர் ஆல் த அமினோ ஆசிட்ஸ் ஆர் அரேஞ்ச்ட் இன் அ சீக்வன்ஸ் இன் அ பர்டிகுலர் ப்ரோட்டீன். ஸோ அந்த மெத்தோடையும் இந்த ஹியூமன் ஜீனோம் ப்ராஜெக்ட்டை நம்ம யூஸ் பண்ணிக்கிறோம். அண்ட் ஹியூமன் இஸ் நாட் பாசிபிள் நம்ம சொன்ன மாதிரி. அண்ட் த குரோமோசோம்ஸ் நம்பர் 1 இந்த மே 2006 தான் கண்டுபிடிச்சிருக்காங்க. ஸோ குரோமோசோம் நம்பர் 1 நார்மலி ஹியூமன் கண்டெயின் 46 குரோமோசோம், அதில் 23 பேர்ஸ். 23 பேர்ஸ்னால் 23 பேர்ஸில் குரோமோசோம் நம்பர் 1 வந்து அதில் என்னென்ன ஜீன்ஸ் இருக்குது, ஹவு த பேஸ் பேர்ஸ் ஆர் அரேஞ்ச் இன் அ பர்டிகுலர் குரோமோசோம் அதை ஐடென்டிஃபை பண்ணது மே 2006 தான் பண்ணாங்க. தட் இஸ் அ லாஸ்ட் குரோமோசோம், தட் இஸ் அ 24th குரோமோசோம். ஓகே, 22 plus XY, that is a 24th chromosome ஐடென்டிஃபை பண்ணாங்க. that is a chromosome number 1. ஓகே, next one is salient features of human genome project. Human genome project, we know that the project is a mega project. It is actually identifying the base pairs in the DNA of human cell. So normally human cell contain various nucleotide base pairs in an orderly sequence. How the base pairs are arranged, in what order they are arranged. For example, adenine thymine, cytosine guanine, adenine thymine, cytosine guanine. So the various base pairs are arranged in some orders. So how this order is taking place in the DNA, in the base pairs. So these analyzing technique only human genome project. And the salient features, why the human genome project are organized, and what is the use of a human genome project. The first one, we identify the 3164.7 million base pairs. Approximately we know 3.3 into 10 to the power of 9 base pairs in human haploid content of DNA. So we identifying the approximately 3164 base pairs in the human DNA in one cell. That the human genome project is works. And average gene, according to the Human Genome Project, average gene contain 3000 base pairs. Normally a human cell contain around 25,000 to 30,000 genes. In that we analyzing the genes means approximately each gene contain 3000 bases. 3000 bases means adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine, guanine, cytosine, thymine, adenine, adenine. Same like that, like 3000 bases are present averagely in a single gene. Some genes contain largest, it is contain 2.4 million bases, that the gene is dystrophin. Due to this affection of this gene only, the genetic disorder myotonic dystrophy may occur, that is actually paralysis of some muscles in our body. So this dystrophin, a gene which contain, a single gene contain 2.4 million bases, adenine, thymine, adenine, thymine, adenine, thymine mari 2.4 million bases are present in a single gene. That is the largest gene present in our cell, that the gene name is dystrophin. The next one is smallest gene, the average gene, largest and 3000 bases, and the 30,000 to actually according to the Human Genome Project we identified 30,000 genes are present in a single cell, but previously imagine 80,000 to 1,40,000 genes are present. In previous report 80,000 to 1,40,000 genes are present, but after the Human Genome Project work is done, the human normally contain around 30,000 genes. In the 30,000 genes, in all the human, whether the human are present in Africa, whether they are present in Australia, whether they are present in Namakkal, all the human contain 99.9% of bases are similar. For example, US human and Namakkal human, the DNA base pairs means 99.9% DNA base pairs are similar. Only 0.1% only is different from person to person in a single species. Species to species may be differ, but in a single species like Homo sapiens, one species. In Homo sapiens, various human are present, various states and various country human are present. In different human, in a single species like Homo sapiens, 99.9% bases are similar. Only 0.1% gene only is different. Okay, then, and normally 100% genes are discovered. The discovered panna genes la 50% genes வந்து function என்னன்னு தெரியலையாம். So we identified various genes, around 30,000 genes are identified with the use of Human Genome Project. இதில் approximately 50,000 genes ஓட function என்ன? What is the function of the particular 50,000 genes, 15,000 genes அப்படின்னு தெரியலங்கிறாங்க. ஸோ அப்போ இது வரல டிஸ்கவர் பண்ணல ஜீன்ஸில் 50% of the genes function is unknown, கண்டுபிடிக்கல, அது முடியல, அது எப்படின்னு தெரியலன்னு சொல்லிட்டாங்க. And the chromosome number 1 has 2,968 genes, அதான் more genes. ஒரு சிங்கிள் குரோமோசோம்ஸில் நிறைய ஜீன்ஸ் இருக்குன்னு சொல்கிறதுல more genes are present in a single chromosome means and the particular chromosome number 1, it contain 2,968 genes are present in a single chromosome. And the chromosome number Y, number 3 XY is a sex chromosome in male, XX means sex chromosome in a female. In chromosome number Y is a sex chromosome in male, contain 231 genes. 231 genes means this is base pairs, not base pairs. The gene means is combination of many base pairs. அப்போ Y geneங்கிறதுல 231 genes are present in a Y chromosome, not a gene, Y chromosome. And 1.4 million basesல single nucleotide polymorphism SNPs அப்படிங்கிறது இருக்காங்க. Single nucleotide polymorphism ஒன்றும் கிடையாது. For example, adenine, thymine, adenine, thymine, adenine, thymine, adenine, thymine, adenine, thymine, adenine, thymine, adenine, thymine. 1.4 million adenine, thymine வந்ததுக்கு அப்புறம் another one base pairs are changed. Example, cytosine, guanine is inserted. அப்போ நம்ம டிஎன்ஏ அனலைஸ் பண்ணினா மேக்ஸிமம் ஜீன்ஸ் வந்து மேக்ஸிமம் பேஸ் பேர்ஸ் ரிப்பீட்டட் n நம்பர் ஆஃப் டைம்ஸ் ரிப்பீட் ஆகிருக்கும். 1.4 மில்லியன் டைம்ஸ் ரிப்பீட் ஆகிருக்கும். ஒன்லி ஆஃப்டர் 1.4 மில்லியன் பேஸஸ்க்கு அப்புறம் ஒரு பர்டிகுலர் பேஸ் பேர்ஸ் வந்து ரிப்பீட் ஆகிருக்கு, சேஞ்ச் ஆகிருக்கு. தென் another 1.4 பில்லியன் வந்து டிஃப்ரென்ஸாக வருது அப்படின்னா மேபி அடினைன் தைமினாக வரலாம். அப்போ ஒரு டிஎன்ஏ அனலைஸ் பண்ணும்போது அடினைன் தைமின் வந்துட்டே இருந்துச்சுன்னா 1.4 மில்லியனுக்கு அப்புறம் சம் சேஞ்சஸ், சேம் அடினைன் தைமின் இருக்குது. அதுக்கப்புறம் 1.4 மில்லியனுக்கு அப்புறம் ஒரு சேஞ்சஸ். ஸோ அப்போ 1.4 மில்லியன்ஸ் ஒன்ஸ் த டிஎன்ஏ ஹேஸ் சேஞ்ச்ட், அந்த பேஸ் பேர்ஸ் ஹேஸ் சேஞ்ச்ட். அதுதான் சிங்கிள் நியூக்ளியோடைடு பாலிமார்பிசம். அது ஒரு பர்டிகுலர் பிளேஸில் அந்த நியூக்ளியோடைடு வந்து சேஞ்ச் ஆகிருக்கு. ஆஃப்டர் 1.4 மில்லியனுக்கு அப்புறம் ஒரு நியூக்ளியோடைடு சேஞ்ச் ஆகிருக்கு. ஈவன் பர்சன் டு பர்சன் இதுவும் டிஃப்ரென்ஸாக இருக்குங்க. இப்போ எனக்கு ஒரு பர்டிகுலர் நியூக்ளியோடைடு சேஞ்ச் ஆகிருக்குன்னா இன் அதர் பர்சன் அனதர் ஒரு நியூக்ளியோடைடு சேஞ்ச், அது பேர் தான் சிங்கிள் நியூக்ளியோடைடு பாலிமார்பிசம். அது ஸ்னிஃப்ஸ். இன் போர்டு எக்ஸாம் இன்னும் லாஸ்ட்டு ஒரு 3 இயர்ஸ் முன்னாடி ஸ்னிஃப் அப்படின்னு கமெண்ட் கொடுத்துருந்தாங்க. வாட் இஸ் மீன் பை ஸ்னிஃப்? SNF is nothing but single nucleotide polymorphism present in a human genes, human genome, human DNA. After 1.4 million times once the DNA, the base pair has changed. And the change of a condition that is single nucleotide polymorphism. Okay, then that is actually the salient features of human genome. Human genome project is a very important project for analyzing the genes present in our cell, present in our DNA. For analyzing these genes, we can identify what are all the useful genes, what are all the harmful genes, how genes are controlling and which inheriting the characters, which gene which controls the character, how the chromosomes contain genes. For example, chromosome number 1 has 2000 genes, up to 2900 genes. Up a single chromosomes la how the genes are located, the distance between one gene to another gene, that is chromosomal map, gene map. So the various research related to the human genome project develop ஆயிட்டே போயிட்டு இருக்கு. Okay, that is the actually salient features of human genome project. The next one is application and future challenge. Application means how the human genome project are useful in other industries. This is actually human genome project is not a simple project. It is with the use of various scientists from different disciplines like microbiologist, biotechnologist, various scientists from different disciplines, various knowledge from different disciplines, modern computers and language which retrieve the system in a system and storing the information and retrieving the information from the system. The human genome project is a very project. It is actually useful in various fields like industries, forestry, agriculture, various fields la use agudhu. For example, human gene irukku. For example, vitamin A gene are present in what, where, that is vitamin A gene are produced in papaya, in mango, in carrot. So we can identify the particular carrot cell and analyzing the genome of a particular carrot cell. Afterwards, which gene we controlling the particular vitamin A production we know. Then the particular gene cut panni with the use of restriction endonuclease, with the use of some enzymes, with the use of biotechnology technique, we cut the particular vitamin A producing gene, and the particular gene we insert it into the rice plant. Means what? The rice plant is producing the vitamin A. With the use of these technique, first we analyzing the what the genes are does, where the genes are present, what are all the genes are present in the carrot cell. So on the genes identify pannada, on the genes enna, on the genes vera enna field la use panna. Maybe with the use of biotechnology we change the particular gene into the rice cell. Means the rice cells normally producing the vitamin A. So vitamin A ங்கிறதுக்காக நீ carrot and papaya depend பண்ண தேவையில்லை. Even normal rice it gives vitamin A. So this knowledge only with the use of genome project, not only human genome project, other organisms ஓட genomes கண்டுபிடிச்சாதான் discover பண்ணாதான் we analyze and use this gene into various aspects, various field like agriculture, forestry, bioremediation, bioprocess, எல்லாத்துலயும் யூஸ் பண்ணலாம். If normally we know the bacteria only decomposing the organisms, decomposing the waste materials. So the bacterium decompose பண்ணுதுனா the decompose பண்றதுக்கு bacterium producing some enzymes. The enzymes means actually we know that is a protein. Protein is controlled by the one particular gene. We know the protein information is present in the gene of bacterial cell. The gene only after transcription process is converted into mRNA. After translation process the gene only converted into protein, that is a particular enzyme. With the use of enzyme only the bacterium it decomposes the food materials, decomposes the waste materials. அப்போ நம்ம bacteriaல எந்த gene and the particular enzymes produce பண்ணதுன்னு தெரிஞ்சிருச்சுனா with the use of genome project we identify the particular gene and the genes எடுத்து other organisms la insert பண்ணினா with the use of biotechnology technique the particular organism which decomposes the substances. But we normally depend on some microbes for decomposing like bacterium and fungi. Suppose and the genes identify பண்ணி முடிஞ்சு அதை எடுத்து வேற organisms la place பண்ணினா the particular organism itself producing the decomposing substances which is act as a decomposer. So appa in the particular genome project idea it is useful in various industries like medicine preparation industries, waste decomposition industries, bioremediation industries, biotechnology, bioinformatics and various fields like genome project ideas are useful. So in future maybe these type of ideas, these type of invention may takes place. Other applications of human genome project as well as another genome project. Okay, yes. Next one is DNA fingerprinting. So DNA fingerprinting is nothing but with the use of DNA we analyzing the individual person identity. So normally with the use of normal fingerprint we identify the particular person because the fingerprint is different from every individual, different from single maybe one human is different fingerprint means another human is a different fingerprint as is a different fingerprint. So with the use of DNA fingerprinting also we analyze the individuality of a person. Is actually DNA fingerprint is normal, is not is actually different from normal fingerprint. Fingerprint is different, DNA fingerprint is different. DNA fingerprinting means in the DNA we know that is 99.9% of DNA is similar in all the individuals of a species, but 0.1% DNA basis is different from one individual to another individual, one person to another person. Only 0.1% is different from one to another person. So this is the base of DNA fingerprinting. அப்போ ஒரு பர்சனுக்கு இன்னொரு பர்சனுக்கும் 0.1% DNA difference இருக்கு. So அப்போ with the use of analyzing the particular 0.1% difference we analyzing the particular person. Suppose in a parental dispute, in a or maternity test, in a person is murdering another person, but we identify the culprit, which person is murdering the particular person. So for this identification the DNA fingerprint is available. DNA fingerprint are useful with the use of saliva. Suppose one person out raping then another person, with the use of sperm we identify the which one is culprit. We identify the with the use of sperm because sperm contain DNA. With the use of DNA sequencer, automatic sequencer analyzer, we analyzing the DNA basis with the use of some examples. For example, we suspect the 5%. So we comparing the 5% DNA, which person is culprit. So easily we identify the culprit with the use of DNA fingerprinting. So DNA fingerprinting is nothing but the basis in the DNA is different from every individual. 99.9% bases are similar to all individual in a population. but in 0.1% DNA basis is different from one individual to another individual. With the 0.1% difference which we identify the particular person. So and the particular person எப்படி கண்டுபிடிக்கிறதுனா with the use of DNA fingerprinting technology. In unique in phenotypic appearance, all the human beings are unique in phenotypic appearance. கொஞ்சம் difference இருக்கும். அந்த 0.1% difference ஒன்றும் இல்லை. Majority of the characters is similar to all individual in a population, but 0.1% difference, color, height, weight, skin tone, various differences are present from between one person to another person. அது phenotypicல ஒவ்வொருத்தரும் தனித்தனியாக இருப்பாங்கங்கிறாங்க. Then sequence DNA is actually danding an expensive task. ஒரு பர்சனுக்கும் DNA